Our next speaker is Lauren Martin. Lauren came to the program with a diverse background in the military, in civil service, coral reef ecology, and finfish aquaculture husbandry. She also came in with a very specific capstone project idea, and she strategically scheduled her year of coursework around its successful execution. This included creating an independent research course to obtain the necessary scientific diving requirements. Unfortunately, due to the public health pandemic, diving was not allowed, and there were many additional setbacks she had to overcome. But with some ingenuity, and by setting up a small genetics lab at her home, Lauren was able to complete a portion of her original project. The title of her presentation today is Finding a Needle in a Haystack, Detecting Abalone Presence Using Environmental DNA.
Thank you, Samantha. If you've ever been diving or snorkeling in the ocean and on a mission to find your favorite sea creature, it can feel a lot like the classic Where's Waldo image. Even when everything seems motionless, it can still be a challenge to find that well-camouflaged octopus or nudibranch that you came searching for. Sometimes you don't find it, but that doesn't mean it isn't there. This is what it can be like trying to find abalone, except you're not confined to searching within one page of a book. You're searching within a vast and expansive ocean. Southern California has 7 species of abalone. They've been fished here for their meat and shell for thousands of years. While once abundant along the coast, abalone populations have been in decline from natural predation, overfishing, disease, and other environmental factors. Both the white and black abalone species have been listed as endangered due to their inability to recover on their own. At a first glance, they may not seem as charismatic as marine mammals, but who could resist saving an animal with a face like that? Population surveys inform conservation managers on how to best protect and restore these endangered species, but abalone aren't exactly easy to find in the ocean. These photos show how white abalone are easily camouflaged by encrusting organisms and algae. The best way to spot them is to look for their light-colored tentacles stretching out from under their shell, and by looking for their respiratory pores. It may be easier to spot the abalone in these photos now that I've pointed them out, but when you're searching for them in the ocean, it turns into a game. Is it an abalone, or is it a rock? In the ocean, most of the time, it's a rock. Since white abalone are found on the ocean floor at depths between 50 to 200 feet, scuba divers can be limited from surveying for deeper populations. And this is where remotely operated vehicles, ROVs, step in. But even without limitations on bottom time or depth, ROVs can't access every survey site due to the risk of cable entanglement with fishing gear or kelp. Both these survey methods are expensive, time-consuming, and they can easily overlook this well-camouflaged animal. This endangered species could really benefit from a tool that is better at detecting cryptic organisms. For my capstone, I wanted to address a new survey method being used to detect rare and cryptic species and how it could apply to white abalone. For my capstone project, I wanted to see if it was possible to detect abalone presence from environmental DNA. Known as eDNA for short, this refers to any type of genetic material from an organism that is left behind in its environment. Sources of eDNA can be cells from skin, feces, scales, or mucus. And similar to genetic traces found at a crime scene, environmental samples such as soil or water can be analyzed for eDNA to inform us of which individual species were present. Using this survey method, we can collect a sample of water, filter the water for cells and DNA fragments, and then extract the DNA. Genetic techniques will then allow us to identify which organisms the DNA came from. Every month, new studies emerge showing how eDNA is more cost-efficient, less time-consuming, and more sensitive than other survey methods at detecting the presence of cryptic species. So to see how effective this method is for detecting abalone presence, I designed a tank experiment to look at how far eDNA could be detected from an abalone under controlled conditions. After that, I collected water samples along the coast to see if we could actually detect abalone eDNA in the ocean. While this may sound like a reasonable experiment that could be conducted at any point in time, the state of California was ordered to shelter in place the week before I was supposed to start my first tank experiment at the NOAA Southwest Fisheries Science Center. As a volunteer there, I was immediately restricted access from the building, which meant I had to come up with something fast or come up with a plan B. I've been planning on doing this capstone project even before I started this program, so for me, there was no plan B. This was the project I was determined to do, but as doors literally kept closing in my face, I started to feel helpless. Luckily, my capstone chair reminded me that the best part about being a scientist is figuring out how to adapt and overcome unique situations. So he sent me home with a genetics lab, and we proceeded to set up the first tank experiment. While we wanted to test eDNA as a survey method for white abalone, we used red abalone as a proxy species to avoid putting unnecessary stress on an already endangered species. For each experiment, we placed a single red abalone in the pictured PVC pipe-framed cage with mesh sides and an acrylic bottom. And in this photo, the abalone is stuck there on the acrylic bottom. This cage allowed us to retrieve the abalone from the bottom of the tank, and the mesh sides allowed for the animal's eDNA to disperse throughout the tank. As a side note, when I say we or us, I actually mean my capstone chair, since he still had access to the facility and was able to set up the experiments and collect water samples for me. This is half of the NOAA Southwest Fisheries Science Center Ocean Development Technology Tank. It holds 2 million liters of seawater, has 2 levels of platforms to view the tank from below, and is one of a kind for testing new technologies. It's difficult to see and appreciate the immense size of the tank from the photo, but it's probably larger than most main exhibits you would see at an aquarium. And this is where we conducted our eDNA experiments. This picture shows the inside of the technology tank from the lowest viewing platform. And circled in white here is the caged abalone sitting on the bottom of the tank about 30 feet below the surface. For each experiment, we let the abalone sit in the tank for 18 hours to allow its eDNA to disperse throughout the tank before we collected any of our water samples. This diagram is a spatial representation of where we collected the water samples in relation to the caged abalone. And while this drawing isn't to scale, I left the objects small to account for the difference in size of the water samples compared to the large size of the tank. Since abalone are benthic and sedentary animals, we initially thought their eDNA wouldn't travel far, so we designed this sampling range within a small 2-meter radius from the animal. Pictured in the upper corner is a Van Dorn-style horizontal water sampler, and we use this device to collect 2-liter water samples from the measured distances in the tank. After the water samples were collected, we coordinated a social distancing handoff, and I filtered them at home using a small vacuum pump and a filtration manifold as set up in the top half of the slide. After that, I turned my office into a genetics lab, and this is where I targeted abalone DNA sequences from the water samples. While this was all done from the comfort of my own home, it also meant that if something broke or stops working, I don't have a backup machine to use, and I have to figure out how to fix things on my own. So whether I wanted to be or not, I am now more intimately familiar with the inner workings of the centrifuge, and I'm happy to report it was a quick fix after taking it apart. For my results, for the first experiment, I collected water samples from these distances from the abalone at the bottom of the tank, and all of the water samples detected presence of abalone eDNA, which is indicated by the green cylinders. And because of this, I expanded the sampling range for the second experiment to see if we could detect eDNA farther from the animal. And here I included surface water samples and samples taken along the far edges of the bottom of the tank. And all of these samples also detected presence of abalone eDNA throughout the tank. For the third experiment, to confirm these results, I also used the same sampling design as an experiment 2. and presence of abalone eDNA was detected in 14 out of 15 water samples collected. The only sample that didn't detect presence is the one highlighted in red on the surface. There's a small caveat to this experiment in that the water sample we collected as a control before placing the animal in the tank showed a very faint presence of abalone eDNA, but the rest of our results were so strong in comparison that we felt it was still worth including in our data. So altogether, these tank experiments show us that a small abalone the size of your palm can disperse eDNA throughout a very large volume of seawater in a relatively short amount of time. And knowing this, there was only one way to find out if this could actually be used as a survey tool. So we took our horizontal water sampler for a dip in the ocean off the coast of Point Loma. And these two sites were chosen based on previous knowledge that multiple species of abalone were known to occur there. Both of our water samples collected from the seafloor showed presence of abalone eDNA. Since we targeted DNA that is found within all local species of abalone, future lab work will confirm which individual species were detected in the water sample at each site. Restoring an endangered species is a lot of work, and it isn't done alone. The picture on the left is a team of divers in an ROV that perform underwater surveys searching for white abalone. The picture on the right is a small portion of the white abalone recovery team that help restock wild populations with captive-bred white abalone. Everyone works hand in hand to save this species. My capstone research shows that not only are abalone capable of dispersing eDNA throughout a large volume of water, but that we can actually use eDNA to detect abalone in the ocean. Researchers that take part in the dive surveys have told me it is really hard to find these animals. My research is a great starting point to incorporating eDNA survey methods to help guide the divers to find more white abalone and help restore an endangered species. A huge thank you to John, my capstone chair. Without him, this project would not have been possible. And thank you to the rest of my committee, Jenny, Marina, and Dick, for all of your support and encouragement along this wild ride. At this point, I can take any questions, Samantha.
Thank you, Lauren. So we have a question from Gretchen in New York, and she asks, do you anticipate any barriers to other scientists using this new method of eDNA detection for abalone detection?
I wouldn't say that there would be barriers, but it would be good for everybody to get on the same page. And right now, scientists are trying to streamline their methods for using eDNA as a survey tool, which is good. So there can be a standard across the board for how we do things, and that can make this survey method more reliable.
And is DNA being used as a survey method elsewhere in the United States?
It's actually being used in broad applications around the United States. Specifically, federal and state agencies have been using it since 2013 to detect invasive Asian carp species in the Great Lakes and the river basins. And soil samples are used to detect invasive snakes in Florida. And this tool is also being used to monitor biodiversity around the states in the water and in soil. So there's diverse applications that have just started to come out within the past 5 to 10 years. And in the next 10 years, I'm sure it will be a standard.
Great. We have another a comment and a question from the web. Neat project is the first thing the person says. This is from Charlotte in La Jolla. And she says, how does your detection limit compare to what wildlife forensics can do from dried shells?
I'm not that familiar with wildlife forensics of dried shells, but using eDNA, we can't detect whether the organism is alive or dead, or we don't know the information of if it came from another source, such as feces from another organism. So these are all things that need to be taken into account when we look at the eDNA molecule from an organism, but if you have degraded shell sources, that would also be a source of eDNA.
Great. And then the last question is from Sarah in San Diego. Will you be able to detect if there's more than one abalone present in an area and how big the area is?
We won't be able to detect exact information, but you can do studies that assess the biomass and the abundance of the animal and the rates of eDNA that you are getting from them. So we can have an estimation from that, but it won't discriminate between biomass or abundance. Thank you.